Number 54. And all this week we're talking about emergent properties that are particular to humans. And in this podcast we're going to talk about how mammals in general maintain body weight. And this is something that I know a fair bit about because this is my area of research. The hormone that we're going to talk about in this podcast is what I've been studying for the last 20 years. It's complex, but I'm going to give you the basics to try to illustrate this property of an emergent property. So there's definitely big diversity in how much body fat a person will maintain. There are the sumo wrestlers here you see on the left that will adopt behaviors that will purposely cause them the maximum weight gain possible. Like they will have a big meal and then go to sleep and avoid exercising when that would actually cause them to lose weight because they want to gain weight so that they can, because it's an advantage in their sport. And then there are individuals who are very lean, very low body fat percentage, and they are trying to carry as little weight with them as possible, like this long distance runner. So there's a lot of human diversity. And then there's also the background of the obesity epidemic. So the Centers for Disease Control, you've heard a lot about them in the last few months, but they have for decades now kept track of human obesity in the United States. And they do that by measuring body mass index in surveys. So body mass index is an indicator of how much body weight or body fat you have. It's not perfect, but it's an indicator. For example, like a super fit athlete with a large muscle mass may have a high body mass index, but they are not carrying a lot of fat. But given that it's an imperfect indicator, and it's easy to report because they can just ask somebody on a survey, how has it changed over time? This is the year I got my PhD, and I'm an old guy, but it's not that long ago in terms of some biological phenomenon. And you have this color-coded map where most of the country is in the 10 to 14% of the population is considered obese. There's a middle sort of diagonal through the country that about 20% of the country is considered obese. Again, this is in 1994, the year I got my PhD. And then now if we skip ahead to how things have changed, there's this animation here from the CDC where you see that there's no blue on this map at all because there's nobody in the 10 to 15% range. They had to change the color code midway through this to add extra categories that didn't even exist when that 1994 map was made. So now, in the most recent data that there are published, which on this map is 2018, there are states where more than 35% of the population is considered obese. So greater than 1 in 3 people are considered obese. This is a real problem because obesity is associated with all sorts of other health problems. And So this sort of phenomenon has happened during my lifetime. I can remember what it was like being a kid in elementary school, and there was one overweight kid in your class. Now, about a third of the class is overweight. You know, if you've ever been to an Ohio, water park in Ohio, you know what I'm talking about. So, what's going on? It's hard to know what's going on, but certainly our control of our own body weight is out of control. And we have this epidemic of obesity. And that word is used a lot these days, epidemic, but it really is a widespread problem that causes long-term health issues, and it's getting worse, not better. So what have we learned about how body fat is controlled? So way back in the 50s, there are some experiments done on rats where they, this is a view of the bottom part of their brain. This is where the spinal cord would come out. And in the bottom part of a rat brain and in your brain, there's a region in the brain called the hypothalamus. And they did experiments where they caused damage to the hypothalamus and then see what happens. They also had these experiments called parabiosis, where they would, it sounds like a Frankenstein experiment, but they actually did this, where you can surgically join the circulatory systems of two rats. So the two rats are sewn together, and the blood from this rat flows into the blood from this rat, and so on. So whatever one rat was making and putting into their blood, both rats shared that. And then they could say, okay, if I damage the hypothalamus in one of these rats, what happens to the other rat? And then see what happens. So then they measure the rats' weights over time. If you do a rat by themselves and you just measure how much weight they gain over time, they do gain weight over time because they grow. They turn into an adult rat. Okay, and that's a sort of a normal wild type rat. And if you hook up two rats together, but you don't do anything to them, you just hook up their circulation, it's not great for the rats, so they don't do as well as a rat by themselves, but they do okay, and they gain weight at a certain rate, not crazy. If you take one of these parabiosis pairs and you look at the average weight of the two rats, if you only damage the hypothalamus in one of the rats, it's significantly higher. And if you compare the two rats and not just average two rats, the one that doesn't have a damaged hypothalamus has a normal weight, a little bit below average, but the one that does have a damaged hypothalamus has an enormously increased weight, almost over 2 times as much as the non-damaged rat. So the hypothalamus is important to maintaining weight. There were known mutations of rats from research labs, commercial labs that provided rats and mice to research communities. So there's a lab in Maine called Jackson Labs. They've been around for a long time, and they are a commercial lab that supplies rats and mice to the research community for experiments. And in their colonies of rats, there had been some spontaneous mutations where the rats were obese. The first of these was called OB for obese, And the next one was called DBDB, for DB means diabetic. And the rat was obese and diabetic. So they took these known mutations, known in the sense that they knew that it made an obese rat, but not known in terms of they didn't know what genes were affected. And what they did was they did these parabiosis experiments. And if you hook up a DBD rat to a wild type rat, the DBD rat increases body weight and fat deposits, and the wild type rat dies by starvation. So even though it's eating, it can't get enough calories in this situation. If you use a different mutation, the OB rat, what happens is the wild type rat in this population, or in this parabiosis experiment, does not change weight, but the OB rat reduces its food intake versus if it wasn't hooked up to another rat, and reduces its blood sugar, and it loses weight. And then if you hook up OB to DBDB, these two different mutations, one is affected more than the other. So these guys slightly gain weight, but these guys massively lose weight. This is a little bit misleading in that the DBDBs are much less affected than the OBs. These guys lose weight dramatically, and they will eventually die by starvation. So these two mutations both end up different both end up in an obese rat, but in different ways. So how are they different? So they took rats and they compared them early in their life and later in their life, and then measured a whole bunch of characteristics of them, their body weight, how much their weight has changed over time, and then how much of that weight was fat. And either in free-feeding mice or pair-fed mice. So pair feeding is you're comparing two mice, like wild type to OB, and you give them the same amount of food. And then you're just saying, okay, what do you do with this given amount of food? Where free feeding, you just let them eat whatever they want to eat. Okay? So if we compare the control, wild type, to pair-fed wild type mice, they're not that different. They gain weight. It looks a lot different, but it's not a lot different. And statistically, because they're not showing you all the error there, and they end up to be about the same fatness, not wildly different. And I'll show you wildly different in a second here. If you compare wild type to OB mice, they are wildly different. So if you look at their percent fat, just look at this here. They start off about 2 times as much fat, and they end up with about 4 times as much fat after 40 days. If you compare wild type to DB mice, they are also much more fat. Not quite as bad as an OB mouse, but still pretty bad compared to a wild type mouse. Okay. So notice here that like at 48, or let's say compare at 20 days old, okay, this mouse weighs about 15 grams. This mouse weighs about 16 grams, so not that different. But if you look at how much of that weight is fat, it's a lot different, okay? All right. If you compare the wild type and the OB mice, again, the OB mice have much more of their body composition as fat. And same is true for DB mice. Okay. Pair feeding versus free feeding. uh, the pair feeding, let's see, pair feeding versus free feeding, they end up being about as fat in either situation. Okay, maybe a little bit worse in pair feeding. Okay, so these sorts of experiments were done throughout the 50s, 60s, 70s, 80s with these known strains of mice and rats that were known to be fat, but again, the genetic basis of why they were fat was not known. And then in 1994, Jeff Friedman at Rockefeller University in New York City, his lab cloned the gene that was mutated in the OB mice, and they called that gene and gene product leptin. Leptos is a Greek root which means thin, and so leptin became the name of the hormone that was missing from these OB mice that would make them thin. And they said, so some of the first experiments trying to say, okay, what tissue is making leptin? And so they did what's called a northern blot, where they look at RNAs from all the tissues. And first, they, just to make sure that the same amount of RNA was loaded in every lane, they put in a detector for actin mRNA, because actin is made in all tissues, and they figured about the same amount of actin is made in all tissues. That's not really true, but it's a close enough approximation, so that if I have an actin band that's this fat in the brain lane, and I have an actin band in this fat in the fat lane, that means the total amount of RNA that I have on this gel is about the same for all these different tissues. And then they specifically looked for leptin, and you can just see just a faint band there. It's made in fat tissue, but no other tissues. Doesn't show up in any other tissues. So you have a gene transcript, the mRNA, that they know this gene is missing in the mice that are obese, and the gene transcript is made in fat tissue, so that seems to make sense. Okay, and then do other animals besides mice and rats make leptin? You're actually looking at this up here, not this down here. So this is called a southern blot, where they take all the DNA out of a cell, and then they cut up that DNA, and then just look for, do you have the leptin gene on your DNA, using a little piece of the gene that is radioactive. Now, the gene is not going to be exactly the same sequence in all these species, and that is why it migrates at different places. But if you see a band here, the answer is supposed to be yes. Now, this is not a great picture because obviously it's in mice, because that's where it was discovered, and it's weak there. But you see it in all. You see a band in all these different tissues. Now, I work on fish. I remember seeing this paper with this blot, and I said, okay, fish have it, and I'm going to go find it. Fish do have it, but it actually took about 10 years for anybody to find the fish version of leptin gene, and that's because this blot is a little bit misleading in terms of how close it is in sequence. But that's a story for another day. But we now know that all these animals have leptin. It seems to be important in the evolution of vertebrates in terms of controlling their body fat. After the original mutation of the OB mouse was discovered, and then leptin was discovered to be the gene that was missing. There was another obese mouse, and they found out there was a mutation in the same gene, but a different type of mutation in that gene. So there are mice that don't make any obRNA, okay, and that's the mutation they have. And then there are also mice that make a ton of obRNA, but they're still obese. So what's going on with those guys? Okay, so this OB2J is that they are these guys are missing the mRNA, but the other mutation, they have lots of RNA. Okay, OB2J makes no RNA, OB makes excess RNA. So these are two mutations in the leptin gene. Both of them result in an obese mouse, but one of them doesn't make any mRNA, therefore it's not going to make any protein, therefore it's missing the signal produced to control body fat. But this one is making more mRNA than usual. So what's going on there? Okay. So how can you make extra obRNA and still be a fat mouse? Well, here's the gene sequence for a small portion of the leptin gene in a wild-type mouse, and here is the gene sequence for one of the mutations where you can see instead of coding for this arginine here, it codes for stop codon, and so it makes no functional mRNA that can be made into functional protein. So it's as if the cell recognizes it needs that signal, and so it's turning on the gene to make that signal, because that transcript does not turn into protein, it just keeps on making the transcript without getting the protein, and so the feedback loop to the gene saying I need more of this leptin is still there, but there's no leptin being produced, and so that's why on this picture here you see a lot of mRNA being made, but this mRNA actually can't turn into protein. Okay, high transcription, no functional protein. All right, so they found a gene that was the candidate that was missing in this obese mouse, leptin. And they found out that this gene product was made in fat tissue, so that makes sense. But what does leptin actually do? So that what they did was they made leptin in the lab by translating the mRNA inside a test tube and making leptin protein. And then they give these obese mice injections of leptin. And when they give them injections of leptin, their food intake drastically goes down and they lose weight. Okay? So if you don't give them injection, nothing happens. If you give them an injection but it's just got saline buffer in it, nothing happens. But you give them injection with this missing protein, leptin, they lose weight and they lose their appetite. Okay? But it doesn't work on the DB mice. Remember, DB stands for diabetic, and they're also obese. If you give them leptin, it doesn't, nothing happens to those guys. So what's going on there? I said before that they lose their appetite, but they also lose body mass pretty regularly. But it doesn't work on the DB mice. So these papers came out in 1994. Everybody was super excited about this because of the human obesity problem. They said, okay, we'll just do this to humans. And humans that are obese, they have a problem with their leptin, so we'll just give them some leptin, and then they'll reduce their body mass, and everybody will be thin and happy. But actually, it doesn't work in humans. And I'll explain why in a second. So, there is a thing called a leptin receptor. So leptin is the ligand that is floating around in your blood. It's a hormone floating around in your blood, just like we saw epinephrine or adrenaline floating around in your blood when you get scared, and then it binds to an epinephrine receptor on your cell. Leptin is floating around in your blood. It's pumped out from your fat tissue, floats around in your blood, and then it binds to a leptin receptor. And the leptin receptor is identified as being in your brain, in the hypothalamic region of your brain. So this is labeled leptin, labeled with a radioactive iodine, and it shows up where you expect, in terms of what we had known about the hypothalamus before in those early experiments, where if they damage the hypothalamus in the rats gain weight. That's where leptin looks like it's binding. Leptin receptor is made all over the body, though. This is an RNA-seq experiment where they sequence all of the RNAs coming out of a particular tissue, and they look for, is there any leptin receptor there? And these bars indicate that almost all tissues have leptin receptors. So almost all tissues are sensitive to leptin. We actually don't really know a whole lot, and there's actually a bunch of controversy about what leptin receptors are doing outside the brain, but we know in the brain, where you can see it's a relatively low concentration, we know that they have big effects on appetite in the brain. So in these experiments, you take a whole brain, you grind it up, and you say, how much leptin receptor do I have in the whole brain? Well, the whole brain doesn't have lots of leptin receptors, but a little part of the brain, the hypothalamus, has a lot of leptin receptors, and so it's just diluted by the rest of the brain, and that's why that signal is low. But nevertheless, there are leptin receptors all over your body, and we don't know what the ones outside your brain are doing yet. People are arguing about that. Okay, so here is how we're thinking leptin works as of 2020. And I can tell you, because I've in this field, since 1994, when they discovered the gene for leptin, there's been 50,000 studies done on leptin. And the reason there's so much interest in that is because of obesity. So if you have something that could work on the signaling system, like a drug, and that could affect how people gain or lose weight, there's a lot of money to be made and a lot of health benefits to be gained by developing an effective therapy. There have been lots of things tried, but they have not worked so far. But still people are trying because, again, because of the potential health benefits, and also because of the money that can be made by a pharmaceutical company. Okay, so the model or way of thinking that we think leptin works, that most people are working on when they're trying to develop these drugs and work on these studies, is called a lipostat. So the idea is that leptin is made by fat tissue, and if you take a wild-type mouse that eats too much, that fat tissue increases because it's storing the excess calories as fat. Fat tissue is making leptin, so more fat tissue makes more leptin, and then that binds to the brain and shuts off the appetite. So here's my model that I think is easier to understand than what's in the book. So here's a real picture of a wild type mouse, and here's an OB mouse, just to give you an idea that they really are fantastically obese. And it works in a feedback loop like you've seen many times in other diagrams of emergent properties. So if this guy here has high fat, so a large percent of its body weight is made up of fat. Fat is the tissue that's making leptin. And so it pumps that high amount of leptin into the blood. That leptin is binding to a leptin receptor. Again, those are all over the body, but the ones we know most about are the ones in the brain. The ones in the brain, when they get a leptin bound to them, turn on genes that turn on your metabolism and at the same time reduce appetite. So it starts a second messenger pathway, just like you saw in epinephrine. Okay, it's not G protein, but it's a similar thing. One thing bumps into another and things get phosphorylated, and they go to the nucleus and turn on genes. And those genes stimulate your appetite, so you're burning more fat, and at the same time stimulate your metabolism, so you're burning more fat, at the same time reduce your appetite, so you're taking in less. If you're burning more and you're taking in less, that means the amount of fat stores you'll have will go down. And fat is the thing that is making leptin. And so if fat goes down, the amount of leptin floating around in your blood goes down. Eventually, it'll go down to the point where it comes off the receptor, and everything that got turned on over here, stimulating metabolism, reducing appetite, the opposite thing happens here. Now, metabolism gets reduced and your appetite gets stimulated. So, high leptin, low appetite, low leptin, high appetite. And just like a thermostat, if the temperature gets too high, the thermostat shuts off. If the temperature gets too low, the thermostat turns on the furnace. This is called an adipostat, in that if your body fat gets too high, it'll shut down your appetite. If your body fat gets too low, it'll turn on your appetite. And so with this sort of feedback loop, you can maintain relatively constant body fat. That's the adipostat idea. Okay. With that idea, what else do we know about leptin? So you don't make constant amount of leptin throughout the day. This is a study of 25 preteen girls or just teen girls, and how their leptin concentrations change throughout the day. So they draw blood at different times of the day. They measure how much leptin is in their blood, and you can see it's not the same throughout the day. So it has a circadian rhythm, like we've been talking about this week. These are called a box and whisker plot, where the average value is a bar. The 50% of the values that are in the middle of the range of the values are in the box, and then the whole range is shown here. So this gives you an idea of not just what the average is, we could have plotted that too, but how much variability there is, okay? And it gives you an idea of what's a real change and what's not a real change. So we know that round about midnight and after midnight, you have your highest concentrations of leptin in a person. So you are not typically hungry when you are sleeping, and so it allows you to get through the night without being woken up by hunger, for example. They did the same sort of comparison, or same sort of 25 girls 6 months later after this study, and then they looked at percent nocturnal increase in serum leptin. So nocturnal means at night, and it goes up at night. And how much does it go up at night? That's on the y-axis. How much does it go up at night in each person? And over those 6 months, did they lose body fat, as indicated by these negative numbers, or did they gain body fat? Okay. And you get this sort of correlation saying that in general, that if you had a big increase at night, then you lost body fat over those 6 months. If you had a small increase at night, you gained body fat. Okay, but you can see there's a lot of scatter around these data, and then you have these numbers here, R, P, and N. So N is sample size. How many girls did they sample? 25. P is probability. You've probably seen this in lab. How likely is it that we would have gotten this result if we had just thrown random data on there? And this says it's very unlikely that only 2 times out of 100 will we expect to see this data randomly. And that's considered significant. Okay. And then the R value has a negative in front of it because the slope is negative, but it gives you an idea of if the relationship were perfect, and that was a straight line, and that we knew that if you have this percent increase in nocturnal serum leptin, you're going to have this much body fat change, and there's no variability, then this R value would be 1. If it's anything less than 1, it gives you an idea of how much scatter is there around that perfect straight line down the middle. So let's look at how you get those sorts of numbers by manipulating numbers a little bit. Okay, so let's take this set of data over here. Okay, here's the Y values here in this column, X values in this column. Okay, and then they're all plotted, and the regression is done, and then says the correlation coefficient is 0.4444. That says we can explain, if we draw a straight line through here, we can explain about 44% of the variation with the straight line. Okay, so now we're going to mess with this data a little bit and see how these values change. Okay. If I bring the data in closer artificially, just to see what is the effect on the correlation coefficient up here, it goes way up. So in this case, we can explain 44% of the variation with a straight line. In this case, we can explain over 80% of the variation with a straight line. So you see there's more variation here, less variation here. If the R value goes up, that means the correlation is stronger and there's less variation. Okay? And then if all of the data fall right on the line, it's almost 100% of the variation I can explain with a straight line. So again, as this correlation value here, this correlation coefficient goes up, there's less variation. Okay? If I change, I still have a high correlation, but I change the value of the slope, that just tells me the equation I would use to predict what the percent change body fat is with percent increase in nocturnal serum level. That would be a different equation, but still highly correlated. And then there are situations where the correlation coefficient is so low that it's considered not to be significant. That this line here is not really that different than a slope of 0, which would mean that there's no relationship at all. Like if I know what the percent increase in nocturnal serum leptin is, that doesn't tell me anything necessarily about the change in body fat. Okay, so there are those situations too. In these sorts of situations where you just, when you're on the edge and of a significant relationship, and you are just just manipulating one data point can change things dramatically. So if you look at this data point here, we're going to get rid of that and switch it out for that data point there. All right, we're just going to move it, from here to there. Okay. And now the slope goes from negative to positive. Okay. Both of those slopes are essentially 0, but you don't want to get fooled by this sort of thing. And this sort of thing can happen where people start throwing out outliers. They say, well, that's a really high value. I'm going to throw that out. There must be something wrong with that. But it can really affect how your analysis goes. Okay, so back to leptin in mammals. And this is one of my favorite pictures on Wikipedia of these OB mice to just give you an idea of how big they are. They sort of sit in the middle of the cage and don't move a whole lot, and they're super unhealthy, and they're hard to breed. You have to breed them with wild-type mice to keep them going. Okay, 50,000 studies later and 25 years later, what do we know about leptin? Okay, we know definitely it is in mammals. It is secreted by adipose tissue. It's not secreted by adipose tissue in all animals, but in mammals it is. In all animals that it's been tested in, it suppresses appetite. So if you give somebody an injection of leptin, whether or not it's a chicken, a fish, a monkey, a rat, a person, it'll suppress appetite. Whether or not it's an lipostat, like I just showed you before, that's what most human leptin scientists still are working with as terms of a hypothesis. But some of the people who study non-humans, like me, are wondering if this is really true anymore. So that idea is still out there. It's the most popular idea, but it's not the only idea that's out there. It does affect metabolic rate in multiple animals. So this has been tried in lizards and in fish and in people and in monkeys. And more leptin metabolic rate goes up, less leptin metabolic rate goes down. Metabolic rate meaning basically how fast are you burning oxygen, how fast are you going through your electron transport chain. This is sort of an interesting twist on leptin in that these OB mice never become reproductively mature. So the males are and the females are sterile. If you give them injections of leptin, so they're missing leptin, if you replace the leptin, they do enter reproductive maturity. In humans, it's been shown that some females who become super lean, like athletes, female athletes who get to below about 6% body fat, they will stop having their reproductive cycles, stop having their periods, and injections of leptin can store that, restore that. And also in lean women who want to become mothers, who are trying to become pregnant and having trouble getting pregnant, there's have some success treating those people with leptin. Also, it's super rare that people have a mutation in leptin, but there have been a few cases where people have a mutation in leptin and they are obese. They also don't enter puberty. They were treated with leptin and their weight normalized and they entered puberty. So it's involved in reproductive maturity in lots of different animals. In these mice that can't make leptin, they also have a compromised immune function, and if you give them injections of leptin, their immune function is restored. There's lots of other things in those 50,000 papers. I'm not going to bore you with all of those. But the bottom line is that it's thought that this hormone evolved as a signal to tell an animal if it's starving. So the story goes sort of like this. If you are out there and you're getting your calories and then you stop eating because you can't find something to eat, leptin will drop pretty quickly in your blood. Concentration of leptin will drop pretty quickly after you start to starve, after you stop eating. And that will tell your homeostatic mechanisms to change their priorities. So if you're starving, it's not a good time to reproduce. If you're starving, let's conserve on how fast we're burning fuel. If you're starving, well, let's not try to fight off all infections. Let's just try to make it through starving, because all these things require a lot of energy, and you want to save the calories you got on board for just trying to survive the starvation event. That's the story. That's the hypothesis about how leptin evolved. And that's what me and a lot of other leptin biologists out there are trying to figure out. What did this thing, how did this thing evolve, and what is it really doing in us?